Cool. There's a handout up here for what we'll get to today if you didn't pick one up.
Yeah, I don't remember. Like after, well, like 2 days. Like for weeks after that, spring, just like
Okay, before we get started, Christina pointed out something on the schedule that I simply wasn't aware of, that September 8th, you have your impact project, okay? And so you're out doing important things. So, not that this is totally unimportant, but you know, there's different things about the schedule that I was given didn't have on it, so we'll figure out how we deal with those lost hours coming up. Thank you for letting me know anytime you see something or know something about the schedule that I might not know. I really appreciate when you bring it up to me very much. So, any questions about anything we've talked about? We kind of started out, didn't go through much material because I want to tell you a little bit about some things that I tend to see in first semester professional schools. Well, we were talking generally just about homeostasis, and it does a lot of wonderful things for us, but out in that extracellular fluid, there's well more than 200 different things that are homeostatically controlled. The body doesn't want them too high or too low. So if something is above the so-called set point or set range, the body has mechanisms to bring it back down to normal. If something is too low, the body has mechanisms to get it back up to normal. And when those mechanisms fail, then we get the signs and symptoms of disease or pathological processes. So when certain bad things start happening, what's really going on is a person's lost some type of homeostatic control in one area, or something is dysregulating homeostasis or challenging it. We'll see some examples of that just very soon. So, you know, things like pH pressure, how much sodium, how much potassium, how much chloride, how much glucose, how much of certain amino acids, they're all regulated. They're all regulated, so you have just the right optimal amount. Now, yes, we're going to see as we go along that there will be times when it's appropriate to go above your resting level. It's appropriate sometimes to go below resting level. That's all part of this dynamic drive. It's not just carved in stone. We will keep plasma glucose at 90 milligrams for every 100 mils of plasma. Sometimes the body needs it higher, sometimes the body needs it lower, and it uses these homeostatic mechanisms to make it just right. So, why on earth is the body put so much into homeostasis? And it's actually very, very simple. It's very simple. That think about proteins in your body. Proteins give your body function. Proteins give you function. And how do proteins work? Proteins, and I don't care if they are muscle proteins, contractile proteins, enzymes, membrane or nuclear receptors or carrier transport proteins or antibodies. Proteins function by sticking or binding to things. And you know, that's really a profound thought if you hadn't really thought about that. But how do proteins work? Well, they either stick or they release. They bind or unbind at appropriate times, and that gives us function. Well, that binding and unbinding is very sensitive to the physiochemical environment, the extracellular fluid. And some proteins, they will only bind if the pH is right. And if the pH then changes, they release. Some proteins will only bind if calcium is just right, or temperature, or whatever. But this binding of proteins that gives us function is regulated by so many of these homeostatically controlled factors. So one of the big functions of homeostasis is to appropriately regulate protein binding and unbinding, and the binding and unbinding gives us function. So homeostasis, protein binding, giving us function. If the binding becomes dysregulated due to loss of homeostatic control, some functional aspects going to be affected. Okay, so, and that's kind of what this says, so to read this when you're studying your notes. So, homeostatic control is regulated by sensor-effector negative feedback loop. Sensor-effector negative feedback loop. So, all these, more than 200 different things that are regulated. They're regulated in a number of ways, but you can break it down and identify sensor-effector negative feedback loops. And so what this means is most of these, well, I'm going to show you the example of glucose because that's clinically relevant. So clinically relevant to so many things you're going to be dealing with your patients, that your body wants glucose right here. Well, there are times like you just ate breakfast. It's going to naturally rise, and your body's going to sense that rise and drive it back down to normal. Sometimes it's too low, and your body wants to get it back up to normal. So there are sensors that sense these variations, and then there are effector processes that make the changes. And once the change is made, whether it's glucose, sodium, pH, temperature, once you get back to that normal range, then things shut off. That's called negative feedback. So this is the way, one of the ways your book shows this. And here is some receptor, and I put in here, you can think of that as a sensor. And these are very specific. There's sensors for sodium, there's sensors for calcium, there's sensors for temperature, but it senses, oh, this is out of range. And it sends a signal which will, one way or another, and there's a lot of different ways that this happens. Anytime information is coming in, that's called afferent. Afferent means coming in, information coming in. And when information comes in saying, oh, sodium is too low, it will set off a series of reactions that flow out. The signal goes out the efferent pathway. And whenever you see efferent, think of information going out. from whatever that integration point is. And that signals certain cells and certain processes do something to help make sodium move to the correct concentration in the bloodstream. And this will go on and go on until you get back to normal. And once you get back to normal, that sensor senses, okay, we're back at normal, and everything shuts down. So negative feedback is once you've gone back to whatever the normal level is, this process of sensing and affecting, it shuts down. The system did its job. So sensory effector negative feedback loops. Sensor, effector, negative feedback. So this is an example right here of a reflex arc. And oftentimes when you think about reflexes, you think about, oh, you go to a, your physician and tap on your knee and you get a patellar tendon reflex. Well, reflexes aren't just involving neural pathways. They do involve neural pathways, but when we see the regulation of temperature, regulation of glucose, those are considered reflexes. And a reflex arc is, so a reflex is a specific, involuntary, unpremeditated built-in response to a stimulus. So this is the great thing about homeostasis and how your body's put together. You don't have to go, sodium's feeling a little bit low, fixed it. Or something like that. You have these several hundred things going on. It's built into this marvelous machine. And, I just tell students all the time that this magnificent machine, I'm absolutely convinced that we probably know someplace between 3 and 5% of human biology. And there's a reason why I and others come to that number. Yeah, there's a whole lot of stuff in your physiology book, but the rate in which we learn new things, new pathways to new molecules that the body utilizes tells us that, and it's always been there. All those pathways and all those molecules that are being discovered literally every week and reported on, they've always been there. So the hard part, building this magnificent machine, that's been done. The easy part, figuring it out, how it works, I don't, we might have 95% of the road left to truly understand it. But all these reflexes, and this is that first slide I showed you, that the body is on automation. There are automatic processes of life. Well, that's what these reflexes are, part of the automatic processes of life. And anytime you see an arc, that means you have to have two limbs to it. There'll be a sensing limb that is constantly monitoring whatever factor, glucose, temperature, pH that you're interested in, and then there's effector processes that fix it if it's too high or too low. So anytime you see arc, it has to have an afferent limb and an efferent limb, okay? So let's look at something here that, unless you've lived under a rock for a long time, and none of you have, you know that you've heard a lot about diabetes in the last 5 to 10 years, right? A lot. So here's the incidence of diagnosed diabetes in the United States. The darker the shade, the greater the prevalence in 2000, okay? Now that's 2000, here's 2010. You notice it got a lot darker in a lot more places. There's a huge public health crisis, and this is being driven by the onset of type 2 diabetes. And we'll talk about many times throughout this semester about the differences between type 1 and type 2 diabetes. So there is this huge increase in type 2 diabetes. And yeah, I just the other day, I got to see the 2015, they don't yet have it in cool charts. So maybe toward the end of the semester, I'll show you the 2015 data. I don't know if those charts and this graph, they're produced by the Center for Disease Control. What do you think that line starting from 1958 to 2010, what do you think that line looks like? What do you think? And it's going to go up. Let's see what it does. Bang! I mean, if this doesn't kind of take your breath away, you might not be in the right profession, okay? There's been this absolute explosion in type 2 diabetes. And you know, lots of times people go, well, maybe the diagnostic markers are better, and maybe this is just an increase in diagnosing the disease. No, the diagnostic markers to really identify diabetes have been around a long time. That doesn't explain it. But there's a lot of new type 2 diabetes cases being diagnosed every year. And type 2 diabetes is, the old term was adult onset. Unfortunately, type 2 diabetes is now being identified in children 8 to 10 years of age. And you know, that's a pretty rough thing. Type 2 diabetes usually takes a decade to a couple decades of bad things happening in your body for it to progress to full-blown diabetes. So I've been in classes where students, before I get to, say, well, what the heck happened in 1997? Something bad happened then. No, it takes decades usually in most people to develop, to go from normal to pre-diabetic to diabetic. So it's what happened in here. And right here, very important date, the Pop-Tart was invented. Right in here, color TV became much more readily available. Right here, the first desktop computers. You know, so there were a number of things, a number of really environmental factors that from here to here was a great explosion in different so-called fast food franchises and their proliferation. So there were a number of things in here that changed our eating habits, our physical activity, our daily physical activity, and you can't really say that there's been a big change in genetics, because there hasn't been, but there's been a big change in environmental factors, and you end up with something like that. Yes?
Did you say that the graph continued to go up when we saw the 2015?
Pardon me?
Did you say that the graph continued to go up?
Something interesting has happened between 2002 and 2015. The steepness is starting to go down. The graph's still going up, but it's actually starting to go down a little bit. That's a really good thing. But because it was actually predicted that out to 2020, it'd probably be on that same trajectory. It's still going up very rapidly, but it's actually starting to do that a little bit. So hopefully, you know, clinicians and people are getting the message. We need to be more careful with what we eat. We need to be a lot more careful in making sure we have enough daily physical activity. So diabetes is the result of a loss of glycemic control. And what is glycemic control? Glycemic control is glucose homeostasis. Glucose homeostasis. So if I'm going to explain to you how homeostasis works, might as well give you a very relevant clinical example. So here we have plasma glucose. Your normal plasma glucose when you're fasting is between 70 and 100 milligrams of glucose in what I did here, in every 100 milliliters of extracellular fluid. There's a lot of different ways it's reported, but it's between 70 and 100 milligrams, or you'll see 70 to 100 milligrams percent, or 70 to 100 milligrams per DL. DL stands for deciliter, and a deciliter is equal to 100 mils. Now there are times throughout the day when your glucose appropriately rises. Let's say you've just eaten breakfast. You've eaten some oatmeal, you drink a glass of orange juice, a lot of carbohydrates that are digested and absorbed as glucose. So after a meal, your glucose might shoot up to 160 to 180. And once it's rising, you don't have to wait until you get all the way to 180 to sense this, but in your pancreatic beta cells, you have a number of different types of cells in your pancreas. Many, many different types of cells we're going to be talking about. Your pancreas is one of your best friends. Be kind to it. But there are sensors. They're constantly sensing, is glucose too high? Now, there is a sensor that senses elevated glucose in pancreatic beta cells. And when they sense glucose is high, what does that do? It causes those beta cells to release insulin. A normal, healthy person, when that plasma glucose is going up, those pancreatic beta cells are going to release insulin, and it's going to go into the bloodstream. And once it gets into the bloodstream, it can go all throughout the body, And it is going to go floating by cells that have a protein. Remember, proteins help give us function. That's called the insulin receptor. And when insulin goes floating by a cell with an insulin receptor, it engages, it binds to that receptor protein, and that receptor sends a signal to the interior of that cell. And many, many cells have insulin receptors. On a mass basis, your adipose tissue and your skeletal muscle, that's where you have most of the insulin receptors. Insulin receptors are very, very important in other tissues, but for what I'm talking about here, the uptake of glucose out of the bloodstream, which we'll go a lot more in detail later on in the semester when we get to endocrinology, where you have the ability to take up most of the glucose out of your bloodstream and skeletal muscle and adipocytes. So when, let's say we are floating past the skeletal muscle that has insulin receptors, insulin binds to it, that signals, that insulin molecule is a signal that says, hey guys, there's a lot of glucose out here in the extracellular fluid. Turn on processes to bring carriers, glucose carriers, to the surface of the muscle and bring that glucose in from the extracellular fluid, from the plasma, to put it inside these muscle cells or these adipocytes or whatever. So insulin signals these cells with insulin receptors to take up the glucose. It's saying glucose is high out here, start bringing it into the cell. And that's what happens. So these effector cells, skeletal muscle, adipocytes, and there's several others, they start taking glucose out of the extracellular fluid. And as they do that, glucose falls from 180 to 160 to 140 to 120 as more and more glucose is taking up out of the bloodstream. And finally, when it gets back to the normal range, that sensor senses, okay, it's no longer elevated, it turns off. That's negative feedback. Negative feedback is when you get back to a normal range and that sensor says, okay, I can turn off. So you have sensor, effector, negative feedback loops. Any questions on that? So we have that for sodium, we have that for calcium, extracellular calcium, we have that for extracellular pH, we have that for extracellular pressure. We have so many of these that will sense. In this case, this pancreatic beta cell has a sensor that detects when plasma glucose is too high. That's its job. And its second job is to tell the cell, okay, release insulin. Why glucose is too high? And then insulin is the signaling molecule to the effector cells saying, okay, take up glucose now. Yes?
Does insulin have a preference for like skeletal muscle cells, like over adipose, or like? Is there kind of a pecking order maybe on which tissues take up glucose the most? There is, but it's not really at the insulin receptor. It's when insulin binds a receptor, it depends on which cell type, but insulin will stimulate dozens of different actions inside the cell. Some of them cause a lot of glucose to come in, some not so much. But
one of your biggest drives in your body is if you happen to have low glycogen stores. Glycogen is chains of glucose molecules stored in your muscles. And if that happens to be low, there's a huge drive for that glucose to go to, during insulin, periods when insulin's high, to go to those tissues that need it first. Now, there are a lot of tissues in your cells that are insulin independent. They can take up glucose without that insulin signal. Thank goodness, your brain and your spinal cord, they don't need insulin. They're constantly taking up glucose. They don't need to wait for the signal. They're just constantly taking it out of the blood all the time. And if any of you know anything about type 1 diabetes, you know that's a great thing, because if the brain was waiting on insulin and an untreated type 1 diabetic doesn't have insulin, brain's off. That person's life's off, just like that, and within minutes. Okay, now, so that's a sensory effector, negative feedback loop if plasma glucose is elevated. Guess what? There is a completely different reflex arc for if plasma glucose is too low. If plasma glucose is too low, pancreatic alpha cells